Rice. Which of the following gene is of bacterial origin? The options are phytoin synthase, caro- carotene desaturase, lycopene beta cyclase, and phytoin desaturase. I'll give you 2 minutes of time. If you know the answer, you can write it in the chat box or you can speak it out aloud. Okay, so the first question is on your screen. In golden rice, which of the following gene is of bacterial origin? The options are phytoin synthase, carotene desaturase, lycopene beta cyclase, and phytoin desaturase. Okay, so let's solve this question. Actually, all these questions and the sessions are mostly the questions which you can remember. They are not much of logical type questions, so not much to explain in this. But okay, let's talk about golden rice first. Okay. So basically, golden rice is a genetically engineered crop, and it is a variety. It's a variety of rice, which is scientific name is Oryza sativa. Okay, and it's a so this golden rice was designed to produce more amount of beta carotene. Okay, beta carotene is another name for vitamin A. Okay. So it was designed in a way that this particular rice variety, the golden rice variety, will have more amount of vitamin A as compared to the normal rice variety or the wild type rice variety. Okay. And this vitamin A is present in the edible endosperm, which is the white part of the rice grains, which is the part that we actually eat. So it became more and more efficient because not just it is getting produced in more amount, but because it is getting produced in the edible part of the plant, it became very beneficial to everyone because you can like we were able to eat our vitamin A in more content just from a small amount of rice. Now, why was golden rice developed? So there are certain countries which rely heavily only on rice, like their staple food is rice. So they because those countries, due to the climate conditions and due to the soil conditions, were not able to grow many other crops, and that is why they were very much dependent on only rice. But rice will naturally lack vitamin A. Okay, so because of their dependency on rice and rice being unable to provide the sufficient amount of vitamin A, this led to high amount of vitamin A deficiency in those populations, and it led to causing diseases like night blindness or weakened immunity or increased child mortality. So now to prevent these things, golden rice was developed to basically address this nutritional deficiency so that those countries which were relying only on rice were now able to eat their staple food and also get the uh, required amount of vitamin A. Okay. So now, what was the change that they made in golden rice? So basically, they wanted to improve the vitamin A content, for which they had to improve the provitamin A, which was the beta carotene content. Okay, and beta carotene content basically will get improved. It has a biosynthetic pathway, and by engineering this biosynthetic pathway into the rice endosperm, there were different genes used to make a genetically modified crop, which was called as golden rice. So what they did was that there is a gene called phytoin synthase. Okay, phytoin synthase or PSY gene, which was taken from plants like daffodil or maize. Okay, PSY gene or phytoindsynthase. Then there was another gene, carotene desaturase. Okay, which is CRT1. So this was taken from Arvenia euridivora. Okay, this is now the name has changed to Pantoia ananatatis. Okay, so this gene was carotene desaturase. The third gene was lycopene beta cyclase. This was again taken from another plant source. So these three genes were basically introduced into the plant for it to help for it to help the plant to generate more amount of beta carotene. And the bacterial gene was basically carotene desaturase, which will encode for carotene desaturase protein, and it will replace multiple plant desaturase steps in a single efficient step. Okay, so now according to our question, okay, so this is a small pathway of how beta carotene is produced. So basically, the initial compound is Geranus genarin phosphate, which is also called as GPP. Then by using phytoin synthase, it gets converted to phytoin. Then by CRTL gene, CRT1 gene, it gets converted to zeta carotene, which then gets converted to lycopene. And by lycopene cyclase, it gets converted to beta carotene, which is our final product. Okay. So now, according to the question, that in golden rice, which gene is of bacterial origin? The bacterial origin gene was carotene desaturase. Okay, because phytoin synthase was taken from daffodil or maize also. Okay. And the other one was lycopene, which was again taken from plant source. Okay. So option B is correct that carotene desaturase gene was taken from a bacterial origin and added into golden rice for it to produce more amount of carotene. Okay. Okay. Moving on to our next question. Which company developed golden rice with 20 times higher beta carotene? Again, this is a question to remember. There is no explanation to this. Okay. So basically, golden rice developed with 20 times higher beta carotene was the another variety which was named as golden rice 2. Okay. The first variety was golden rice 1 and this was named as golden rice 2. Now, basically, the improved version or golden rice 2 or GR2, which will produce up to 20 times more beta carotene, it was developed by scientists in collaboration with Syngenta company. Okay, they replaced the phytoin synthase gene or the PSY gene from daffodil with the maize PSY gene, and which that led to a production of much higher amount of keratin accumulation, and this also significantly enhanced the nutritional value as compared to the first version of the golden rice. Okay, as you can see in this, these are some images of golden rice. This first one is the wild type variety or the normal rice with which lacks uh, huge amount of beta carotene. Then this was golden rice one, which had the PSY gene from daffodil, and this is the another variety, GR2. in which the PSY gene was from maize. It had more amount of beta carotene content. Okay. So the correct answer to this is option A, Syngenta. Syngenta developed golden rice variety with much higher amount of beta carotene content. Okay. So the next question is, which of the following enzymes is responsible for conversion of lycopene to beta carotene? The option is carotene desaturase, phytoin desaturase, carotene desaturase, and lycopene beta cyclase. Okay. So let's talk about this question. Okay. So the question says enzyme responsible for conversion of lycopene to beta carotene. Okay. So we already talked about this pathway in the previous question. The first compound that was made is from GPP. It gets converted to phytoin by synthase, then gets converted to zeta carotene, then to lycopene, and from lycopene to beta carotene, it gets converted by the enzyme called as lycopene cyclase. Okay, so basically, this enzyme converts beta carotene. This converts lycopene to beta carotene. Okay, so in this carotenoid biosynthesis pathway, basically lycopene is a linear molecule and it is converted to beta carotene by enzyme lycopene beta cyclase, which will add a beta ion ring at both ends of lycopene molecule. Okay, so the roles of all the other genes that was mentioned in the option was carotene desaturase is involved in earlier desaturation step. Phytoin desaturase will convert phytoin to carotene, and carotene desaturase will convert to intermediate desaturation step. Okay, so according to our question, that which gene converts responsible for the conversion of lycopene to beta carotene, the correct answer is lycopene beta cyclase. Option D in this case is correct. Okay, so the next question is which of the following is initial precursor molecule of beta carotene? The option is isopentyl pyrophosphate, geranyl diphosphate, phytoin, and D is lycopene. Again, you can answer this question by the previous pathway that we have been talking about in all the questions. Okay. So let's solve this question. So again, beta carotene is synthesized using this isoprenoid pathway or terpenoid pathway. And in this case, the basic building block of all isopropanoids is basically isopentyl pyrophosphate, okay, also called as IPP. So this compound basically starts the pathway of terpenoid or the isoprenoid pathway. Now, in this case, let's this is the simple pathway sequence where IPP gets converted to DMAP, then it gets converted to GGPP, then to phytoin, then to lycopene, and then to beta carotene. So basically, IPP here is the initial precursor molecule, but this geranyl diphosphate, it is the immediate precursor of carotenoid synthesis. Okay, this particular compound is the immediate precursor. So in this entire isopropanoid pathway, if the question is asked, you need to understand certain terms which will change the answer. Okay, so if the question is that in this beta carotenoid pathway or the terpenoid pathway, which is the initial precursor molecule, then the answer will be IPP. If the question is asked that what is the immediate precursor of carotenoid synthesis pathway, then the answer will be geranyl diphosphate or GGPP. If the question is which is the first committed compound, committed carotenoid to the step, then the answer will be phytoin. That means that once phytoin is formed, then that means it will definitely get converted to beta carotene. There is no other step or no other rerouting that the compound can take to form any other compound. Once it gets formed into phytoin, that means beta carotene is definitely going to get produced. So it is the first committed carotenoid in this entire pathway. Then then the question is that what is immediate precursor of beta carotenoid? Beta carotene. Then the immediate precursor of beta carotene is lycopene. Okay. So the terms used in the question will determine what the answer is. So if the question is being asked at earliest precursor, then IPP is the correct answer. And if the question is asking which is the direct precursor, then lycopene will be the answer. This is the small pathway you can like remember to answer these kind of questions. That in native pathway, first acetyl coenzyme A is converted to HMG coenzyme A, which gets converted to mevalonic acid, then gets converted to IPP. So from here, the terpenoid pathway will start. Now this IPP gets converted to FPP and then to GGPP, which is geranyl pyrophosphate. Now from here, it can either get converted to phytoin. So as I told that this is the committing step. Okay. Once this phytoin gets formed, that means beta carotene is definitely going to get formed. It gets converted to lycopene and lycopene gets converted to beta carotene. FPP can also form squalene or ergosterols. Okay. So this is the pathway that you can remember to answer this question. Now the question was asking about which is the initial precursor molecule of beta carotene. Okay, so initial precursor molecule here is isopentenyl pyrophosphate. That is option A. If the question would have been for like the immediate precursor of the carotene pathway, then it would be geranyl diphosphate. Or if it is about the immediate precursor, then it will be lycopene. Okay, so it's talking about initial precursor molecule for beta carotene. So option A, isopentyl pyrophosphate is correct. IPP. This is IPP, and this one is GGPP. Okay, all right. So the next question is which selectable marker gene is used in golden rice 2? The options are NPT2, APH4, PMI, and HPT2. Okay. So let's answer this question. We already talked about golden rice 2, okay, or GR2 variety. So the selectable marker gene in the GR2 variety is the PMI gene. Okay, it is phosphomannose isomerase. Now, BMI gene will allow the transform cells to grow on the media, which will contain mannose as their carbon source, and it is considered as more safer and non-antibiotic selectable marker, which makes it more acceptable for regulatory approval of a public acceptance because we are talking about a plant variety which is supposed to be consumed by humans in later time. Okay, so that is why it is it requires a more safer and more like non-antibiotic selectable marker, so that there is no chance of uh, by mistake the gene which is antibiotic or any harmful gene getting introduced in the plants that is being developed. Okay, so that is the PMI gene, which is phosphomannose isomerase gene. It will help the plant to utilize you to basically the selectable marker, which will help to utilize mannose as its carbon source. Again, why others are not used? Because NPT and HPT both are hygromycin resistance. They are antibiotic resistance genes, and APH4 is also hygromycin B resistance gene. So all these are avoided in developing a variety which is supposed to be edible. So that is why PMI gene is used. This is the gene structure of golden rice too. You can see here that this is the PMI gene. Okay, and PMI gene, uh, and it has a NOS terminator. So this will basically help in selection of the transformed variety. Okay, so the correct answer is option C, PMI gene. Okay. Next question is, which type of promoter is used in golden rice for PSY and CRTI? The option is endosperm specific promoter, fruit specific promoter, root specific promoter, and anther specific promoter. Okay, so let's solve this question. In this case, the question is talking about the um, promoter which is used in golden rice for PSY and CITI gene. So in golden rice, the goal was to produce more amount of beta carotene. Okay, and not just producing more amount of beta carotene, but to specifically produce it in the endosperm, which is the edible part of the rice, so that beta carotene or the vitamin A can be consumed upon eating. So therefore, in this case, the PSY gene or the phytoin synthase gene that is being used, and the CRTI or the keratin desaturase gene that is used, they are placed under the control of endosperm specific promoters. The reason being that we want that particular gene to get expressed in the endosperm so that it can come to the edible part of the rice. So, when placing it under the control of endosperm specific promoters, it will ensure that beta carotene will accumulate only in the grains and not in any other parts of the plants, because we don't want that. Because even if beta carotene gets produced in other parts of the plants, it won't be consumable and it will not be of our use. Okay, so that is why the targeted expression in endosperm was done to avoid unnecessary metabolic burden on the plant or the other tissues, and it will ensure nutritional enhancement where it matters the most, right? So the correct answer to this question is A, option A, endosperm specific promoters. Okay. So the next question is Bacillus thuringiensis was discovered as a pathogen in which of the following? The options are cotton bollworm, flower moth, luna moth, and clothes moth. Okay, so let's talk about this question. It's The question talks about Bacillus thuringiensis was discovered as a pathogen in which of the following? So, if you all remember, we have talked about Bacillus thuringiensis in previous sessions, but as a protective mechanism, as BT cotton as a gene which was taken from Bacillus thuringiensis, and it was incorporated into cotton plant to prevent it from pathogen attacks. Okay, but originally BT was a pathogen. Okay, which was infecting insects, and from there, scientists were able to find out that why and how, basically, what was the mechanism which was helping this particular Bacillus thuringiensis to basically kill the larvae. So to understand that, so that's how they understood the mechanism, and then they isolated that particular gene which was responsible to kill the larvae and transformed it into the plant, which was later on used by the plant to protect itself. But now the backstory of that is this question, that Bacillus thuringiensis, in which basically insect was it discovered as a pathogen. So the correct answer is flower moth to this. Okay, PT or Bacillus thuringiensis was first discovered as a pathogen for infecting the larvae of flower moth in 1911 by Ernst Berliner, and he was the first one to isolate the bacterium from this diseased flower moth in Germany. Okay, basically Thuringia is a place in Germany from where this particular Bacillus thuringiensis got its name from. So if you look at these photos here, so if you talk about this first photo, so in this case, this first larvae is how the normal larvae of flower moth grows, but this upper one is infected by the bacillus. Okay, so that's how it will kill the bacteria, and then this is the normal larvae, and how once it is infected, it will basically the gut and everything will come out, and it will basically kill the larvae. In D1, it is the moth that is getting affected. So if you look here, that how the effect of Bacillus thuringiensis shows at a later stage of uh, the in the later stage of this moth. Okay, this is the small basic life cycle of Bacillus thuringiensis, how it affects basically. So first, the spores of this Bacillus thuringiensis is ingested by the target insect, and then the spores will get dissolved in the gut, and where it will form the active toxin, which is, if you remember, that cry protein is produced. Okay, cry protein is produced by the cry gene, and this cry protein will basically uh, um, come into its active form, and it will disrupt the gut of the larvae. And once it disrupts, it will form more and more pores, and then that's how the insect will die. Okay, so this is the life cycle of how Bacillus thuringiensis basically affects the larvae. So the correct answer to this question is that BT was first discovered as a pathogen in flower moth. Option B is correct. Okay. So the next question is, a primer pair for a gene is designed. Choose the strands 5 dash or 3 dash of the template DNA where the forward and the reverse primer will bind. Okay. So these are the options, that forward will bind to 5 dash to 3 dash, and reverse will bind to 3 to 5. Forward will bind to 3 to 5, and reverse will bind to 3 to 5. Forward will bind to 5 to 3, and reverse will also bind to 5 to 3. And forward will bind to 3 to 5, and reverse will bind to 5 to 3. This is a logical question. Like, you need to understand the concept of how primers bind to the template. I'll just give you 2 minutes. You can try solving. It's not that difficult. Okay. So let's solve this question. All right. So basically, DNA polymerase will synthesize the DNA only in one direction. Okay, this is the well-known fact that it will always synthesize the DNA in the direction of 5 dash to 3 dash, right? So that, okay. So the primer should always bind on the strand in such a way that the extension should proceed in the 5 dash to 3 dash direction, right? Now, for the DNA polymerase reaction to work, and if the primers are designed, that means there are always two primers. One has to be a forward primer, and the other one has to be a reverse primer, right? So now, because there are two primers, and because the direction has to be only 5 dash to 3 dash, that means that both these primers will bind to two opposite strands of the DNA. Now, suppose if these are the two DNA strands, and one strand is 5 dash to 3 dash in this way, and the other one is 5 dash to 3 dash, right? These are the two DNA strands. Now, if we are talking about the primers, so we know that the direction of synthesis will always be from the 5 dash to 3 dash. That means the primer binding will always happen in the direction of 5 dash, and it will extend up to 3 dash. So one primer is supposed to bind here. Okay, let's change the color for this. So one primer is supposed to bind here, and one primer is supposed to bind here, right? Now the question is that which one of of this will be forward and which one of this will be reverse. So now, uh, in this case, the direction in which the extension is going to be in the same direction of the 5 dash to 3 dash, or the strand is basically the strand is 5 dash to 3 dash. That is where the forward primer will bind. So the forward primer will have the same sequence as the coding strand, and it will bind to the antisense strand, or the strand that will run from 3 dash to 5 dash. Okay, that means this strand, this one. And the other reverse primer, it will bind to the complementary strand, or it will bind to the coding strand. And because it is binding to the coding strand, it will run from the coding strand will run from 5 dash to 3 dash direction. Okay, so now the answer to this question is option D, that forward primer will bind to the 3 dash to 5 dash direction, and reverse primer will bind to the 5 dash to 3 dash direction. Okay, so this will be your reverse primer, and this will be the forward primer. And the synthesis will happen in this direction, and here the synthesis will happen in this direction. Okay, so I hope this is clear that how you will figure out that when the two primers are binding forward and reverse, in which direction it is going to get extended. Okay. All right. I hope this is clear. Okay. So the next question is, in flavor saver, the shelf life of tomato was increased by which of the following gene silencing techniques? The option is A, CRISPR, B, RNA interference, C is antisense RNA, and D is AMI RNA. Okay. So let's talk about this question. So this question talks about a variety of tomato, which is called as flavor saver tomato, and this is also a genetically modified variety of tomato. And this was developed to increase the shelf life of tomato and to reduce the amount of time that it increase the amount of time that it takes to get completely ripened, so that the rotting will basically take a longer time and the tomatoes can be saved for a longer time. Okay, so now, okay. So basically, flavor. Give me a minute. Okay. So basically, flavor saver tomato was first commercially approved as a genetically modified food crop, and its shelf life was increased by suppressing a gene which is called as polygalactourinase. Okay, or the PG gene. Now, PG gene that produces this PG enzyme. Now, this PG enzyme is responsible for the fruit to become more soft. So, the more the fruit will become soft, the more it will ripen, and more easily it will get rotten. Okay, so to prevent this, this flavor saver tomato variety was developed, which will suppress or decrease the production of this PG enzyme. Now, how this was done? So, this was done by basically using antisense RNA technology, where antisense copy of the PG gene was introduced. So, if this is the PG gene, okay, so PG gene will produce uh, basically an mRNA, okay, which will be a PG mRNA. And this will convert into protein, right? Or the PG enzyme. And this enzyme will play a crucial role in softening of the tomato, right? This is how the mechanism works. But in case of antisense RNA technology, there is another antisense PG gene introduced, which will form the reverse mRNA. Okay, it will form an RNA which is antisense RNA. Okay. Now, what this antisense RNA will do? That this antisense RNA will bind to uh, the original RNA that is produced in the plant. Okay. And now, by the mechanism of dicer and argonaute protein, this entire mRNA will get cut into small interfering RNAs, and the enzyme that is being produced will now not get produced. Okay. This enzyme will now not be produced. And because this enzyme is not produced, softening will not happen. Okay. That is the working mechanism of antisense RNA technology and how the flavor saver tomato was made. That is exactly given here, that antisense RNA will bind to the normal RNA, prevent its translation, and then reduce the enzyme production, which will lead to delayed softening. Why other techniques were not utilized? First of all, CRISPR was not available at the time when a flavor saver tomato was formed, so it was not developed at this time. RNA interference, it is conceptually related to flavor saver tomato, but it is not the exact method that was utilized. And AMI RNA is advanced RNAi based technique. It was not developed at this time. Okay, so this is the mechanism, that in case of normal tomato, by a figure you can understand it better, that a normal mRNA or the sense mRNA is produced, which will get converted to protein, and it will produce normal PG and lead to natural rotting. But in case of this transgenic or the flavor saver tomato that we are talking about, in this case, along with the normal or the sense mRNA, there is another antisense mRNA which is produced by a synthetic antisense gene. Sense mRNA is produced by the PG gene, which is already present in the plant, but the antisense mRNA will be produced by synthetic antisense gene that has been introduced. This will lead to complementary mRNA formation. This will get degraded and not form the PG gene. Therefore, ripening and rotting will not happen for about 3 weeks. Okay, this is the working mechanism of flavor saver tomato. So now the correct answer to this question, that which technique was used, gene silencing technique was used for it, correct option is option C, or the antisense RNA. Okay, so the last question is why BT toxin does not affect cattle and humans. The options are: toxin ingested by cattle or human is inactive due to heat. The toxin is inactivated by alkaline pH, which is unavailable in human or cattle. The toxin is impermeable in mammals, and the toxin is degraded by the saliva. Yes, so let's talk about this question that why BT toxin does not affect cattle and humans. Now, why, first of all, why is it important that BT toxin should not affect cattle or humans? The reason is that when we are making a genetically modified crop, which is going to express a gene that is capable of killing a larvae. And the working mechanism is also interestingly that it will affect in a way that it will disrupt the gut lining. Okay, so now if that protein is also able to do the same thing in the humans or in the cattle, that means that the crop will become first of all inedible for humans, as well as it will be poisonous to the cattle, that if they are growing and if some cattle by mistake feeds on that crop, it is going to have a harmful effect on the cattle as well, right? But that is not the case with BT toxin, that it does not affect the cattle and humans. Now, why? So, to answer this question, you need to understand the mechanism of how BT toxin works, right? So, BT toxin is obtained from Bacillus thuringiensis, right? Bacillus thuringiensis will produce a toxin which is called as crytoxin, and it is a now when BT will produce this crytoxin, it is basically in its inactive or the protoxin form, right? Now, how it becomes active? When this protoxin or the inactive form of crytoxin enters inside the insect larvae, the insect gut basically has an alkaline pH which can range from up to approximately pH 9 to 10. Right now, this protoxin or the inactive form of toxin, for it to get converted to active form of toxin, it requires a low pH, right? The pH should be low for it to become come into its toxic form. So protoxin gets activated when the pH is low, which is available in the gut of the insect larvae, and this is when the active toxin will bind to the gut receptors and it will form pores and the insect will die. Now, in case of humans and in cattle, the stomach environment is acidic or the pH is low. So, because the pH is low and it is not getting this condition to get converted to protoxin, to get converted from protoxin to active form of toxin, that is why in humans and cattle, protoxin does not get activated and it gets digested like any other normal protein and gets excreted out. So the key concept here is that Pt toxin specifically depends on the alkaline gut pH and specific receptors which are present inside the insect midgut, which is not present in humans or cattle. Neither the pH is available nor the specific receptors are available. Okay, this is a figure that will help understand better that when from leaves, if the ingested bacterial Bacillus thuringiensis spores are ingested, it has to get converted to the protox. It has a protoxin form which will require a certain pH. Now, when it gets activated, the active toxin form will basically bind to the receptors in the gut lining, and it will increase the permeability, and the pores will start forming in the gut. And when the gut lining will start having this multiple pores, the insect is going to eventually die. This is the working mechanism of BT toxin, and because this pH here is not available in humans and this binding receptors are not available in humans or in cattle, that is why this entire mechanism does not work on humans, okay, or on cattle as well, right? So the correct answer is option B, that is toxin is activated by alkaline pH, which is unavailable in humans or in cattle, and that is the reason why BT toxin does not affect cattle or humans. Okay, I hope this concept was clear. If there is any doubt, you can ask. And if no, we can end this session. And this was the last live interaction session for this particular topic. And all the very best for your exams.